So by introduction to metabolism, that word metabolism is really, it's an everyday word that we use, although it's not usually used properly. So when we use the word metabolism kind of commonly, we're using it to refer to something that has to do with usually weight loss. So people talk about whether they have a fast metabolism or a slow metabolism. But the word metabolism really is much more inclusive than just, say, the reactions that break down fat molecules or utilize the food that we consume. So by metabolism, we're talking about all of the reactions that occur within an organism. So the totality of an organism's chemical reactions. This is going to include different types of reactions. These will be both catabolic and anabolic pathways. And you'll see in just a little bit the definitions of those more specifically. But really we're talking about both reactions that would break down molecules. Those will be our catabolic reactions. And then also reactions that are going to build complex molecules. So we're really seeing both ends of the spectrum. Now, if you look at this diagram right here, I know you can't really see the reactions that are on here, but what we're looking at is a picture of all of the reactions that take place within a cell. Now, we're not necessarily getting every single reaction every moment of every day, but the big idea with this picture is that there's definitely a lot going on inside of a cell. So if we just zoom in on a little area here so you can get an idea of what we're looking at, the point is to understand that not only are there lots of reactions inside the cell, but there's also a lot of connections between these reactions. So when we talk about pathways, a lot of times we think of them or we look at them as linear, isolated pathways and reactions. But in reality, they are connected pathways. There's lots of forks in the pathways. So we have decisions that are made all of the time inside the cell, basically to decide what a cell is going to do with the limited amount of energy that they have. They will certainly not be able to do everything, so they'll have to make choices as to how to spend that energy. So in this slide, we're seeing the way that pathways are generally studied. And this is the way that we're going to look at them in the next two chapters. Essentially, you have a molecule, we'll call it molecule A here. That molecule is going to get converted into molecule B, and we will have one enzyme that's going to make that reaction happen for us. Then we'll take B and we will convert it into C, utilizing a second enzyme. And then C will get converted into D, utilizing a third enzyme. So all in all, we're going to get molecule A converted into D, and it's going to be a multi-step reaction involving multiple enzymes. Now keep in mind, though we look at it like this, there are forks in the pathway. So it may be that sometimes B actually gets converted into, say, E. Or maybe sometimes we convert it into F or some other molecule. And so when a cell actually has B to work with, it has to decide, do I need D, do I need E, or do I need F? And which one should I convert it into? And those would all use different enzymes. So keep in mind these pathways are interconnected. Now, if we talk about different types of reactions, we mentioned catabolic and anabolic pathways on the very first slide, and these are basically opposites of each other. If we're talking about a catabolic pathway, this is defined as a pathway that breaks down complex molecules into simpler compounds. An example of this would be taking something like a starch molecule and breaking it down into the component monosaccharides that make it up. And we could also take those monosaccharides and break those down further as well. Now, the reason that catabolic pathways are run is because they are going to release energy. And if they release energy, essentially that energy can be used for other things. A big example, or a good example, of a catabolic pathway is going to be cellular respiration. Now we're going to look at cellular respiration in a lot of detail in Chapter 9, and that is the focus of Chapter 9. Cellular respiration is the most prevalent catabolic pathway that we know of that takes place. So it really is a very good example for us. Cellular respiration is the process that we use when we consume food molecules of different types, whether it be protein, fat molecules, or sugars. It's how we break down those molecules and actually get energy out of them. Now, the other type of pathway is going to be an anabolic pathway. Anabolic pathways are essentially the opposite of the catabolic pathways, and that these are pathways that build complicated molecules from much simpler molecules. When they do this, because they're building very complicated structures or molecules, they will require or consume energy to make that happen. Now, anabolic pathways we will look at in chapter 10, and that will be photosynthesis as far as our big example of an anabolic pathway. So photosynthesis is the subject of chapter 10. In photosynthesis, basically plants and a few other organisms are able to take small molecules like carbon dioxide and basically assemble them into sugar molecules that can be utilized by the plant or utilized by other organisms. Because they are building complicated molecules, they will require energy for that, and that energy is going to come from sunlight. So that really is why plants are going to require some light. So we'll be looking at both of these types of pathways in detail in later chapters. In chapter 8 here, we're interested in really the definitions and understanding what happens to the energy in both pathways. Now, because we're talking about energy, we need to first go through again and define energy. This study of how organisms manage their energy resources is called bioenergetics. So bioenergetics is what we're starting to touch on here. Now, energy, we have defined it previously, and there are a lot of different definitions that you could use here, one of which would be the capacity to cause change. Oftentimes you see the ability to do work. Kind of like the second one here as it applies to the chemistry that we're going through. It is also defined as the ability to rearrange a collection of matter. When we talk about chemical reactions, we really are just rearranging a collection of matter or moving the atoms around, maybe breaking some bonds and reforming some bonds in a new arrangement. We do have two types of energy. Those are the kinetic and the potential. Kinetic energy is really an energy of motion or movement. And then when we're talking about potential energy, that's energy that is stored up. And really that stored up energy, as we defined in chapter 2, is going to be due to a set of matters, basically position, location, structure. So every piece of matter that we have is going to have some inherent potential energy because it's going to have some kind of a structure to it, and it's also going to have a specific location. Now, a type of energy that we'll be talking about a lot in this chapter and in the next few will be chemical energy. Chemical energy is potential energy, and this is potential energy that is stored within a molecule that is going to be available for release in a chemical reaction. Now, this diagram from your textbook just more specifically looks at energy and just kind of defines it or makes sure that we understand the different types of energy. We talked about potential energy quite a lot in chapter 2 with regards to the atom structure. And we discussed how electrons, depending on their location, have different amounts of potential energy. Basically, the further out they were from the nucleus, the more potential energy they had. And then the closer in they were, the less potential energy that they had. Notice in this diagram, we have a man standing on a diving board. And because he is, say, up high on that diving board, he is going to have more potential energy than, say, when he is actually sitting in the water. So the higher up he gets, the more potential energy he would have. The lower he is, the less potential energy he would have. Keep in mind that if that diving board is not physically holding him up in the air, then he is going to fall into the water. So basically, all living matter really wants to get to that low potential energy state, as we described in chapter 2. Now, if he were to jump off the diving board, here we say the man actually, we see him actually going down. That would be kinetic energy that we're seeing. So that would be energy of motion. So another thing to keep in mind is that when he's up on the diving board, we could say that he is less stable. or we could say more unstable, and less stable is going to be more potential energy. Once he is down here in the water, we would say that he is more stable. Basically, he doesn't have very far that he could actually go. And the more stable something is, the less the potential energy that it's going to have. It's very important that we understand the correlation between stability and the amount of potential energy that something has. So again, the more unstable or the less stable it is, the more potential energy we have, and the more stable, then the lower the amount of potential energy that we have. If we introduce thermodynamics, this is a study of energy transformations that do occur within a collection of matter. It does apply to the reactions that we have take place inside the cell. So we do want to introduce a few of the laws of thermodynamics and understand them so that we do keep them in mind as we talk about our chemical reactions. Now, as far as some terminology, no matter what, when you're talking about science, you do have some kind of matter that's going to be under study. Now, that could be something that is living or non-living, but that is what we call our system. Okay, so if we're studying a plant, that would be our system. If we're studying a rock, that would be our system. The surroundings are like the environment. So the surroundings would be everything outside that particular system. Now, systems themselves are classified as either being closed or isolated systems versus open systems. And this classification has to do with how those systems interact with their surroundings. A closed or isolated system, those are not going to have an interaction or an exchange of materials with their surroundings. And then an open system is going to be just the opposite, because an open system will have an exchange of materials with the surroundings. In biology, we're really interested in living organisms, whether they be bacterial cells or plants, humans, animals of the other types. And anytime you have a living organism, that living organism is going to be an open system. Okay, so all living organisms will be open systems. Keep in mind that we are constantly consuming food. We're constantly releasing waste. We are inhaling air, we are exhaling air. So that is definitely an exchange of materials with our surroundings. Now, if you consider if we stopped breathing, so if we stopped taking things in from our environment, taking in air or taking in food from our environment, then we would rather quickly die. So if a living organism is not in an open system, they are going to die. The closed or the isolated systems, these are going to be our non-living things. Okay, so this would be something like a rock, a book, a pen, something of that sort. Now, students often get confused if we talk about taking a living organism and we stick it into some kind of a container or cage. So a fish in a bowl, for example. If we have a fish in a bowl, a lot of times students want to call that a closed system. It's still a living organism and it is still interacting with the surroundings. So if you take that fish and you put it in a bowl, you will still be adding food to that bowl. There will still be oxygen available to the surface of that water in the bowl. So there will be an exchange of materials there between the fish and the environment or the surroundings. So just because we put it into a container or a cage of some sort, we do still want to keep in mind that that is an open system. Now, if we go to our laws of thermodynamics, the very first law states that energy can be transferred and transformed, but it cannot be created or destroyed. So that means that we have a set amount of energy on the Earth's surface, and we're not going to be gaining new energy on the Earth's surface, and we're not losing any energy on the Earth's surface. So if I need energy, for example, I can get that energy, but it's not going to just come from nowhere. So I can consume some type of food. So I can take the energy out of the candy bar and consume it, and then that's transferred to myself. So we can definitely move energy from one place or one object to another. Now, when we do that, that's where the second law comes into play. So every time we have an energy transfer or transformation, that will increase the entropy of the universe. Entropy is perhaps a word that you haven't heard of before, and entropy is a measure of disorder or randomness. So if we are transferring energy from one place or one object to another, which we do all of the time, then we will get an increase in the entropy or the disorder of the universe. So this means that over time, the universe is becoming much more disordered. Now, if we look at these two laws in regards to some pictures that you have in your textbook. This first picture here is showing something similar to what I described to you with me consuming food. So in this case, we have a cheetah. He needs some energy. So he is consuming another animal that had that energy. So he's getting that energy in the form of fat molecules, protein molecules, carbohydrates of some sort. So we are transferring the energy from one animal to another. Now the second law there in the second picture said that every time we have an energy transfer, then we are going to increase the entropy or the disorder of the universe. So in this picture, that cheetah is using that energy to actually run. So we're transforming it into, say, a kinetic energy. As we do that, we take molecules that were once nice organized molecules in the form of a protein or a fat molecule for that matter, and we're breaking those molecules into small pieces such as carbon dioxide and water. So we're breaking it into a bunch of scattered little pieces, and at the same time some of the energy is going to be released as heat energy as well. So by taking something that was once a nice organized molecule and breaking it into little bits and pieces, that is a description of increasing the entropy in the universe. Now we're going to be talking about chemical reactions. We're talking about chemical energy, which is a potential energy. And we're going to be very interested in how the amount of energy within a molecule changes from the beginning of the reaction to the end of the reaction. So we're going to call this the free energy change. And where this term free energy comes into play, this was defined by a man with the last name of Gibbs. So we do call it Gibbs free energy, and we represent it with a capital G. This Gibbs free energy is the portion of a system's energy that can perform work when the temperature and the pressure are uniform throughout the system. In other words, this is basically chemical energy. It does certainly apply to a living cell. And if we look up here at the Gibbs this free energy change is represented by a triangle or a delta, which is a Greek symbol, and then it's going to be the free energy change. So we're going to be talking about a delta G. The delta G is the change in energy or the free energy change, and the value of this free energy change is going to determine whether or not a reaction will occur spontaneously. So whether this will be the natural tendency of something, as we talked about with, say, passive transport, versus whether or not we'll have to really force something to take place, like active transport. In this slide, the first equation here shows you some of the components that go into figuring out the free energy change. There are a number of components in there. We're not going to talk about using that particular equation, although if you go on to take physics classes, you're very likely to see that equation. We will, though, be using the second one here, which states that the change in free energy is equal to the amount of energy in the final state, or you can think of this as the amount of energy in the products, subtracting off the amount of energy in the initial state, or you could think of it as the reactants. So if we look at these three examples, we're back to our diving board example. And as we said before, if the man is on the diving board, we would say that he's going to have more potential energy, or we could say a higher G. More G, higher G. So more free energy up there. Once he jumps off and he lands in the water, he is going to have a less amount of free energy. So a lower G for that matter. Now, if we were to just throw in some numbers here for illustration purposes, if on the diving board he has more, we'll say he has 10, and if after he jumps and falls in the water he has 3, Now we've got some numbers so that we can plug something into this equation that we've got on the top. So if we take the amount of energy in the final state, that would be 3 in our example, and we subtract off the amount of energy in the initial state, that would be 10, and when we work that out, we end up with a -7. Now, what's important to us in this chapter is not the 7, but it's the fact that that number is actually negative. So if you have a negative delta G value, as we do in this first example, then that will mean that that's going to be a spontaneous reaction. In other words, if that diving board does not physically hold him up, he will fall to the water. So that's the natural tendency, is that he will fall right into the water. If we look at the next example, this example is diffusion, and we said that diffusion was the natural tendency of molecules. They wanted to spread out or go from an area where they were very crowded or in a high concentration to a low concentration. This applies to any molecule. If we assume that, say, the molecules in this diagram happen to be cations, that would put a little positive charge on each one of these molecules. And we know that like charges repel each other. So if you cram a bunch of these positive charges in a tight confined area with each other, they will repel. They're going to be very unstable in that tight confined space. Keep in mind that we said that if something is unstable, it's going to have more potential energy, or more G, higher G. If these actually spread out, as we see down here, they're going to not be repelling each other with the same amount of force. So this is going to now be more stable, and more stable means that they have less G, or a lower amount of energy. Again, if you threw numbers in here, you would end up with this reaction having an overall negative value. So by it having a negative value, that means that the molecules want to go from very concentrated to spreading out or being less concentrated. And that's why diffusion was actually passive transport. In the last example here in C, we've got a complex molecule that is going to be broken up into smaller bits and pieces. Now you can think of this similar to the diffusion example, because if we have this complicated molecule, that complicated molecule is going to really be constrained in movement. We've got a lot of atoms that are in very close proximity to each other, and their electrons as well. So if we break this up into smaller pieces, everything kind of just has more free range of movement. It's going to be more stable. Now what we're looking at here is really a catabolic pathway. When we describe catabolic pathways took complicated molecules and they broke them up into smaller bits and pieces. Catabolic pathways will also have a delta G value that is going to be negative. Now keep in mind that if we have a system at equilibrium, the G, or the free energy, will be at its lowest possible value in that system. Previously, we said that all matter wants to proceed towards low potential energy. So by proceeds towards low potential energy, that's really saying the same thing, to the lowest potential energy, or the most stable state that's possible. If you think about diffusion, we said that diffusion is going to occur until we get those molecules as spread out as they can possibly be. So if they are as spread out as they can possibly be, that would be that most stable state. Now we do have two different types of reactions depending on the delta G values that we get. These reactions are called exergonic reactions or endergonic reactions. Now these are very similar to some terms you may have heard of in previous classes, so exothermic and endothermic. When we use that thermic term, it really specifically applies to heat energy. Here we're just talking about energy in general. So if we look at the exergonic reaction first. This diagram here is showing us how an exergonic reaction changes in the amount of energy that we have. Now on the y-axis we have the free energy, so that we could label as G if we wanted to. And so the higher a bar is, the more energy that substance has. And if you look at the x-axis, we have the progress of the reaction. So notice that the first bar represents the reactants. In this case, they happen to have quite a lot of potential energy, or a lot of G. And the products would be over here towards the end. And in this case, they do not have as much energy. In this reaction, we start with a reactant that has a lot of energy, and we end up with a product that has a lot less. This is what we get when we have an exergonic reaction. Now, if we start with a reactant that has a lot, and we end with a product that has a lot less, and we think about that first law of thermodynamics, that law stated that the energy is not simply going to disappear. So this decrease in energy that we have comes about because energy is going to be released. So as we see this little arrow point out, that's saying that energy is being released in this process, which is where the term exergonic comes from. So by exergonic, we're saying energy outward, or energy is coming out of this particular reaction. We could put some numbers on this scale over here. We could say that this is going to be 3 and this is going to be, say, 10 up here. And we'd have the same example we looked at before. So if we plug in some numbers, the final state, or the products, actually would have 3. And the initial state has 10. And overall we end up with a negative number. This difference here that we see on our chart is the actual delta G. In this case, with the numbers we've plugged in, that delta G is really equal to a -7. But the important part is to understand that it is negative. If you have a negative delta G, that means that you do have an exergonic reaction. By exergonic reaction, we are going to have a release of energy. It will also be spontaneous. And if you keep in mind the two types of pathways that we initially described, the catabolic pathway is going to be exergonic. So we want to keep in mind that all of those terms kind of go together. Exergonic, energy outward, a release of energy, spontaneous, negative delta G, and catabolic. As well, if you saw a diagram as we see on this slide, we would want to be able to state that that is an exergonic reaction. And if there were numbers on the y-axis, we would want to be able to come up with a value for our delta G. Now if we look at the flip side of this, here we have a diagram representing an endergonic reaction. Endergonic reactions are really just the opposite, because notice in this case we start with a reactant that has a lot less. We'll use the same numbers here and say that it has 3 as far as its potential energy, or free energy, goes. And we have a product that then has a lot more. So we'll give it 10. Well, if we take these numbers and we plug them in up here, now we've got a final state that has 10, and we've got an initial state that has 3, and we're going to get an overall +7. So when we talk about endergonic reactions, we're talking about reactions that have positive delta Gs. Now, because we're going to end up with a product that actually has an enhanced amount of potential energy, we do actually have to put energy into this. So notice that our energy is really going in. So this is where the term endergonic comes from, energy inward. And this is going to mean that this requires energy. This is going to be a non-spontaneous reaction. By non-spontaneous, this means that if we do not have the energy to actually put into this reaction, the reaction is not going to take place. Just like active transport. In active transport, we said that molecules go from a region of low concentration to high concentration. They don't really want to move in that direction. But we were able, in active transport, to actually force them to move in that direction if we had energy to put into it. So endergonic reactions require energy. They're going to be non-spontaneous, and these are going to be our anabolic pathways. All right. So we do have two types of pathways: exergonic and endergonic. We want to know the difference between the two. Now, if we go back to our idea of closed systems, we said that living organisms cannot be closed systems. And this diagram right here is illustrating the reason for that. We said that all reactions really want to proceed towards the lowest possible state of potential energy, or lowest possible state of free energy. That's equilibrium. So in the first diagram, we see water flowing down this hill. As it goes down the hill, we've got the wheel turning and we've got a light bulb on. So things are happening in that particular system. Now, if there is no water coming in and there's no water going out, eventually we're going to get to the second picture here, where we see that the water has evened out, the wheel has stopped spinning, and the light bulb's not on. That would be where we're actually at equilibrium. Now, if we're at equilibrium, basically nothing is happening. So for a living organism, equilibrium of all of the reactions would be a dead organism because nothing is happening. So we never want to get to equilibrium. So if we look at how this would be modified to describe an open system, with open systems, we are interacting with our surroundings. So this means that there are inputs coming in and there are outputs as well. So notice now we just have some water coming in and then we also have some water leaving. If that is the case, then that wheel is going to continuously spin and will constantly have the light bulb on. Now, if we look down at the second picture, that really is a better description of what we have taking place inside the cell, because it's not going to be one isolated reaction. Instead, we're going to have a lot of different reactions. So as we described initially, we said A maybe gets converted into B, and maybe that gets converted into C, and maybe that gets converted into D. Notice that what comes out of the reaction of A to B is actually going to serve as the input for the next reaction, and so on. So these reactions are basically feeding one into the next, and way back we do have some inputs into our body, and at the very end we have some outputs there as well. So open systems will be living organisms. Now cells are going to need basically 3 different types of work to keep them running. One of those will be mechanical work. Mechanical work would be some type of movement work. This could be movement of an entire cell, or it could be movement within the cell. So if we're talking about, say, a vesicle moving from, say, the endoplasmic reticulum to the Golgi apparatus, that would be work. We also have transport work, which we described in chapter 7. So things like active transport. If we have a need to accumulate a large concentration of a particular molecule, we would have to utilize some energy to make that happen. And we also have chemical work that needs to take place. So by chemical work, this would be the making of different molecules, synthesizing proteins, making phospholipids for our membranes, things of that sort. Now, because we have these 3 different types of work, inside the cells we will be utilizing a process called energy coupling. Energy coupling is basically going to take an exergonic process and it's going to use it to power an endergonic process. So these 2 reactions are really going to take place side by side or hand in hand with each other. So when we think about the exergonic process, the exergonic said that those released energy, always. And we will be running exergonic processes inside of our cells because they will release energy. And then we can use that energy to power the endergonic reactions. Because keep in mind, the endergonic reactions, those actually require energy. Okay, so we run the exergonic process because it's going to release and provide some energy that we need to force our endergonic reactions to actually run and take place. Now, the molecule that's going to be involved in energy transformations for the most part inside the cell is what we call adenosine triphosphate, and this is ATP. We saw a little bit of ATP being utilized in chapter 7. When we saw it in chapter 7, it was like a little starburst of energy, and at that point we just said this is energy that's being utilized. But if we look at the structure of ATP, ATP You don't need to know the entire structure, but we do want to focus in on this part that's shaded tan here, because this is really the critical important part of the molecule with regards to its function and being able to store energy. Notice that we've got 3 phosphate groups in a row. These phosphate groups happen to have lots of negative charges on them. And as we said previously, like charges are going to repel each other. So what we've done on this end of the molecule is we've taken a bunch of negative charges and we've crammed them in quite tight proximity to each other. This means that this tail is going to be highly unstable. And if we have a tail or this end here that's highly unstable, this means that this is very high potential energy, or high G. So we've got a lot of energy held within that tail. Specifically, this energy is going to be held in the bonds between the phosphate groups. If we were to cut some of those bonds there between the phosphate groups, that would enable the negative charges to spread out some. And if they spread out some, then we would end up with a more stable molecule than we had before. So if we look at an example of this, the reaction that you're seeing right here, this is cutting with water. So this is a hydrolysis reaction. In this hydrolysis reaction, we're going to take water. We're basically going to force it right in between those 2 phosphates. So between, say, the second and the third phosphate. When we do that, one of the phosphates will come off, and we'll call that one inorganic phosphate because it's no longer connected to carbons and hydrogens. And then we also have what's left with 2 phosphates on it, which we call ADP. And the DP stands for diphosphate because we only have 2 of them now. Now, if we look at this kind of on an energy diagram as we looked at before, we're going to have our energy over here on this side, or our free energy. This would be the progress of the reaction on the bottom. So we're going to start with a reactant, which is ATP in this case. This ATP we said is highly unstable because of those negative charges that are shoved together. We're going to clip off one of the phosphates in this hydrolysis reaction, and we're going to end up with a product which will be ADP. Now ADP will not be as unstable because it doesn't have as many negative charges crammed together in tight proximity. So if you look at this, this really is an exergonic reaction that we're looking at here, because we're going to start with a lot of energy in our reactant right here, and then we're going to end up with a product that has a lot less in the form of ADP. So as this reaction proceeds, there will be some energy that's released in the process. That's the energy that we see right here. ATP for cells will be used as a short-term energy storage molecule. So what that means is that when we need a little bit of energy, we'll basically clip one of those bonds in the ATP molecule, and energy will be released. And then the cell will basically grab that energy and use it to run some endergonic process inside the cell. Now keep in mind that we can have ATP, which actually has 3 phosphates. Okay, so that would be my ATP. We can also have ADP, which would have 2 phosphates on it. And it is possible to have 1 phosphate attached as well, which is going to be AMP, which would be monophosphate. Now, if we talk about the amount of energy in each one of these molecules, the AMP is going to have the least amount. And that's because you only have 1 phosphate group there, so we don't have all of those negative charges crammed together. The ATP is going to have the highest amount of potential energy because you have all 3 of the phosphates with their negative charges next to each other. In the middle, okay, would be your ADP that has a medium amount of potential energy within it. Now, if we actually do have ATP hydrolysis, we said that ATP hydrolysis is going to be an exergonic reaction. We can put an actual value on that. It is a negative delta G because it is an exergonic reaction. So we see a negative 7.3, and unit-wise, it's going to be kilocalories per mole. This means that when we have that process take place, this energy that is released is in the quantity of 7.3 kilocalories per mole. The importance of ATP hydrolysis is that we can take that 7.3 and we can use it to run other reactions. And those other reactions that we will be running will certainly be endergonic reactions. So if we look at how this is going to be utilized inside the cell, we do pretty constantly need to run some endergonic reactions. So in this first example here, we see this glutamine, which happens to be an amino acid, actually being synthesized. Since it is an amino acid, it's going to be required to make proteins, which is something we pretty steadily need to make inside the cell. And notice that since we're really synthesizing or making something, it does have a positive delta G, and it happens to be 3.4 is the actual number. Now the fact that it's positive means that that is a non-spontaneous reaction. So in other words, if we don't have the energy to put into that reaction, it's never going to happen. So this is where ATP hydrolysis comes into play. So it's a pretty complicated diagram here, but if we look down here at the bottom, if we take ATP hydrolysis and we split the ATP apart, as we said before, we get a negative delta G, and the value is actually 7.3, negative 7.3. So 7.3 kilocalories is being released when we run that particular reaction. Well, the endergonic reaction that we want to run in this case actually requires 3.4. So 7.3 is obviously greater than 3.4. So if we can actually add those numbers together, and we can come up with an overall number that happens to be negative, that means that these two reactions, if we run them side by side, they will both take place. So we're going to use ATP hydrolysis to provide the energy to run the first reaction. Now, what we're seeing here is basically that this reaction usually takes place in kind of a two-step mechanism. So when we talk about ATP hydrolysis, there can be multiple steps involved. We'll talk about that multi-step process more in chapters 9 and 10. The important thing to keep in mind here is that ATP is useful to provide the energy to run endergonic reactions. It's not just going to be any endergonic reactions, but it would be endergonic reactions that are going to need less than 7.3 kilocalories per mole. If we have a reaction that has, say, a delta G of for instance, plus 10, we're not going to use ATP hydrolysis for that because 7.3 is not plus 10. So obviously breaking apart an ATP would be a little wasteful there because it's not going to be able to cover the amount of energy that we need. ATP is a useful molecule inside the cell because it has an ideal amount of energy inside of it. It provides just the right amount of energy to run a lot of different reactions inside the cell. As we saw down here in this example, you do have some extra left over in this reaction. So we have an extra 3.9 in this case. Well, when energy is released during a reaction, it is a burst release of energy. And this means that whatever energy is going to be captured or utilized has to be captured or utilized at that particular instant. So we really will use the 3.4 to run the endergonic reaction, and we really won't be able to capture, we won't be capable of capturing that 3.9. So it's going to be lost pretty quickly, usually in the form of heat from the cell. So we don't really want to use a molecule that has tons of energy inside of it, because if we did use those molecules, we would be losing even more energy as heat. Here's just some other examples showing how we would use ATP to run, say, transport work or mechanical work. Transport work, active transport. We looked at that in chapter 7. And then mechanical work we haven't really looked at in detail, but it does require some energy to actually have some movement take place. So here we see some ATP being used in the movement of a vesicle along a track. In all of these cases, it's ATP hydrolysis that's being used. So we're taking ATP, We're breaking it into ADP and phosphate, and in the process of that, we do have energy that's going to be released. It's important that we understand that ATP is renewable inside the cell. In other words, we have a set supply of ATP components. Sometimes those components will be hooked together in the form of ATP. Sometimes they will be broken apart into ADP and phosphate. So we've got a limited amount to work with, and we'll constantly be basically hydrolyzing ATP. And when we do that, we're breaking it apart into ADP and phosphate, and then we're going to want to put it back together again. If we look at these two reactions, we looked at the ATP hydrolysis already, and we said that it had a negative delta G. So that means that energy is released in that case. It's exergonic. On the flip side of this, if we need to actually put the ATP back together, putting it back together or making ATP is going to be endergonic. So that one actually has a positive delta G. So if we we hydrolyze ATP, we break it apart, and energy comes out. This energy is used to do some kind of work inside the cell. But then we're left with the components of ATP, and we need to stick them back together again so that we can use ATP again. So in order to get this side to actually happen, we said that this would be making ATP. This is going to mean that this is going to be anabolic or have a delta G that is positive. So in this case, we're going to need energy. That energy is going to come from catabolic or exergonic processes. So we constantly consume food to get some energy. That food is going to be broken down in catabolic pathways, and we'll get energy out. What we will specifically use that energy for is to put the ATP back together again. Then we'll use that ATP to release the energy that we need for other processes inside the cell, and we're going to be constantly doing this. So keep in mind, we constantly have energy coming in because we're constantly eating, and then we're always going to have energy basically going into other reactions inside of our cell. So whether they are transport work, mechanical work, or we talked about the chemical work. So transport, chemical, and mechanical work. Make sure we understand which of these two reactions is endergonic and which one is exergonic. So the making of ATP is going to be endergonic, and that one will require energy. And the hydrolysis or the breakdown of ATP is going to be exergonic, and that one is going to release energy.